My talk for today's webinar is harnessing protein conformation to power antibody discovery. This is focused on engineered cell surface proteins as rational immunization designs. For today's presentation, I will include 5 different sections. The first section to introduce a Conigen briefly, and the second section to talk about the importance of proteins in antibody discovery. The third section will elaborate soluble dimeric proteins. The fourth section will talk about multi-pass proteins displayed on viral particles. The last section will talk about GPCR antibodies. Conigen is a young, innovative life science company based in Worcester, Massachusetts, very close to Boston. So we have extensive expertise on membrane proteins and antibody development. For the membrane proteins, we are focusing on design and produce engineered conformational surface proteins, include 2 different platforms. The first platform is the single-pass dimeric proteins for those natural dimer molecules. The second platform is multi-pass transmembrane domain proteins. It's focused on the full-length transmembrane domain proteins displayed on viral particle. The third platform is also very important to develop antibodies against the challenging targets. So here, give a brief introduction about the 2 protein platforms. The first platform is focusing on the single transmembrane domain proteins. Those proteins form natural dimers on the cell surface. So those natural dimer proteins on the cell surface include many receptors. For example, cytokine receptors, growth factor receptors, immune checkpoints, T-cell antigens and B-cell antigens, tumor-associated antigens, and also other dimer molecules, for example, integrin. And also, the second platform focuses on the more complex multi-transmembrane domain proteins. So these multi-transmembrane domain proteins, we displayed using nanomolecular particle. So these proteins, including 2 domains to 14 transmembrane domain proteins. For example, GPCRs, ion channels, transporters, those are all belong to this multi-transmembrane domain complex proteins. The third platform is also important to generate antibody responses against the difficult targets. When we're talking about therapeutic targets, majority of them are endogenous antigens. Unlike viral proteins or bacterial proteins, those endogenous proteins has high homology between human and other mammals. So therefore, it's very difficult to generate antibodies in laboratory animals because the high homology causes immune tolerance. And also those proteins are complex. The conformation and also the structure is not easy to display. So in this platform, the goal is to break immune tolerance to generate antibodies against those challenging targets. So for this platform, we have 3 key components. The first component is the fit-for-target immunogen design. So in this one, we'll first to use our Conigen's proprietary algorithm to analyze the target, to design the antibody, the protein sequences to mimic the natural epitope and the conformation. Also include the immune engaging element and also some other elements to prolong the antigen presentation in the inside the in vivo, inside the immunized animals. So based on the target, we also select immunogenic format, so specifically for that target. So we know every protein is different. Even the protein in the same category or same family, they are different. So we have to design the unique immunogens and also the forms of antigen protein for that specific target. The second element is the proprietary molecular adjuvant formulation. So this formulation can attract monocytes, antibody presenting cells, antigen presenting cells, those to engage the innate immune response, and also to promote the B cell responses, including B cell activation, B cell survival time, and the maturation to generate high quality antibody responses. The third component is called the immunofocusing. The immunofocusing immunization regimen design basically is to focus on the targets, not other proteins, to generate antibody against the target specifically and with high quality. So here's the 4C of the Conigen's GPCR antibodies generated in the last half a year, a very short period of time, covered a broad range of targets. These targets including different classes based on structure for class A, B, C, and F, and also cover different therapeutic areas. This is the most important, the 10 therapeutic areas all covered by the target we selected. The next section, I'm going to elaborate the antigen and the proteins, the importance for antibody discovery. So since the monoclonal antibody was discovered in 1975, that was a landmark for the antibody world. So since then, in the last 50 years, the antibody technology, especially monoclonal-based therapeutics has blooming and expanded rapidly. And also it's one of the major biologics expand very rapidly and quickly. So the first proved therapeutic antibody was in 1986. This is called antibody called OKT3, targeting the CD3 molecule for kidney transplantation to suppress the immune response. Since then, the antibody, especially monoclonal-based therapeutics, has been evolved a lot in the last 40 years. So including the therapeutic antibody forms, antibody discovery technologies. The key thing is that the monoclonal antibody produced the specific therapeutics, and also is a very essential tool for research and diagnosis. And these antibodies has make the biology and the therapeutics advanced a lot in the years. So this is the one to showing since the first antibody, therapeutic antibody was approved in 1986, in the last 40 years, there are over 200 therapeutic antibodies approved worldwide. Those antibodies cover different therapeutic areas and also have different antibody forms. The initial antibody is called the naked monoclonal antibody, IgG, and then further develop the antibody with other forms, including bispecific antibodies, antibody drug conjugates, ADCs, and then the CAR-Ts, and also some other combined antibody therapeutics. If you look at the left panel of the graph, to date, the naked monoclonal antibodies still the majority approved antibodies. However, all other form antibodies also grow very quickly. The red panel showing the therapeutic areas. The first antibody is targeting for transplantation. Since then, the antibody therapeutic areas has expanded to almost all the kind of diseases, from cancers, infectious diseases, and also many of the chronic diseases, including cardiovascular diseases and also autoimmune diseases. So we have heard many successful stories for antibody discovery, for therapeutic antibodies, and also antibody technology development. So one of the cases is the antigen, proteins, immunogens. This is I think it's a centric of antibody discovery. Although the antibody advanced for the technology make the antibody discovery more efficient. However, if without the antigen, without the protein, the antibody discovery will not be successful. So this is the one showing the proteins are foundational for antibody discovery success. So here is showing the antibody discovery platforms in the years, from traditional hybridoma to phage display, yeast display, humanized transgenic mice to generate antibodies, and also single B-cell-based technologies, including FACS-based and also fluidic-based technologies. And also the most recent development, AI-driven de novo antibody designs. So for all these technologies, all need a good quality proteins. Without the good quality proteins, the antibody discovery will be hurdled. So the proteins can be used as immunogens to immunize animals to generate antibody, and also can be used as antigens for antibody hit selection, and also epitope mapping and antigen that can be used for many other applications as well. So we all know the proteins has 4 levels of structure: primary, secondary, tertiary, and quaternary. So all these structure levels are important to the protein functions. However, so the tertiary structure and the quaternary structure, those are more from the conformational epitopes. Those more directly responsible for each of the protein's specific bioactivities and functions. And also, if we look at the approved therapeutic antibodies, majority of the antibodies targeting membrane proteins and also targeting the conformational epitopes. So however, so to generate these antibodies was not that easy. So in the past, the majority of the antibodies generated using target expressing cells. So now we need new modality of the proteins that can mimic the native conformation to further advance the antibody discovery. So therefore, the protein conformation is critical for antibody discovery success. So for the protein forms, there are commonly used proteins, including peptides, monomeric proteins, cells, viral particles, and in recent years also use genetic immunization, including DNA immunization, mRNA immunization, to generate antibody responses. If you see the protein forms, there are many choices. However, there are few good options. For example, the peptide. The peptide is easy to produce. However, the peptides usually does not have conformational epitopes or more linear epitopes. And also the peptides are not immunogenic. Most of them are heptans. So the heptans have to conjugate to a carrier protein to generate antibodies. When you use the carrier protein, sometimes, majority of the time, to generate antibodies against the carrier protein overcome to the target antibodies. The monomeric proteins, if the protein is naturally a multimeric protein, this protein will lose the quaternary structure and important conformations. For the cells, there are also many other proteins in the cell usually can generate antibody responses against many other proteins and so on. So therefore, we need better proteins and advanced toolboxes for antibody discovery. See, here is a successful story, antibody discovery against RSV. This is the viral infection diseases. In 2023, an antibody called nirsevimab was approved. This antibody was very efficacious and also have good safety profiles. The antibody was generated using a stabilized protein with a conformational structure. This is just one of the examples. On the left panel, if you see the crystal structure, the bottom showing the pre-fusion RSV fusion protein trimer. The top showing this antibody bound to the trimeric protein, the interface. So this antibody was generated use this stabilized pre-fusion trimer. So from RSV infected adult donor memory B cells. So this is a result indicate that this story indicate that the conformation is very important, and also quaternary structure is important for therapeutic antibodies. At Conigen, we use the stabilized interferon alpha receptor 1 and 2 heterodimer and the generate antibodies against the dimeric interface. So these details will have a follow the detailed discussion. The next section, I'm going to discuss the Conigen soluble protein dimer platform. So this protein dimer platform focuses on the extracellular domain of the single transmembrane domain proteins. They are naturally dimer. So in the nature, there are many proteins that form dimer. So based on proteome, there are over 2,000 type 1 transmembrane domain proteins in the protein genome. And also over 300 of them are receptors. Majority of them form dimers. So based on the PubMed publications, in the last 40 years, the research focus to the dimers has dramatically increased, and also more discoveries of dimeric proteins. So there are many studies has shown that the receptor dimerization is a universal mechanism for cell signaling and also for cell functions. And also this is preformed dimers and also the lag-induced dimer conformation are all important to trigger the cell signaling and all the signal process. So in the past, because the technology limit, many of the proteins we thought they are monomeric proteins. With the technology and the scientific advance in the last decades, more and more proteins were identified as a dimer instead of monomer. Here are the example is PD-1. PD-1 and the receptor PD-L1 or PD-L2. In the past, we all know they are monomer proteins. However, the recent study demonstrates that those are also form cis dimers on the cell surface. The other one example is the B7 protein families. So they also form dimers on the cell surface, then also important for their functions. For example, B7H3 is an important therapeutic target. On the cancer cells, majority of these molecules can form dimer. So the dimerization can be a potential good target to target for antibody discovery. So in the nature, there are many dimers. So if we simplify to clarify these dimer proteins, we can, based on the structure, to classify as preformed dimers. That means these dimer receptors, they are naturally formed them on the cell surface. There are many of the examples. For example, the molecule on the CD3 epsilon delta, and also the ligand-induced dimers. For those things, dimers, basically two molecules close to each other when bound to the ligand to bring these proteins form dimer. This is called ligand-induced dimer. Based on their activities or functions, we can also divide into physiological dimers and the pathological dimers. The physiological dimers, there are many of them. So the cytokine receptors, the majority of them form dimer on the cell surface and the natural condition. The pathological dimers, those can form dimers during the diseases and cause different stage of disease progression. Also, there are many examples. One of the example is the alpha-synuclein or neuron degeneration. The first, they will form dimers and higher order oligomers. Another example is the CD117. It's sometimes very difficult to differentiate the physiological dimers and the pathological dimers. CD117 is an example. Naturally, it's formed dimer by ligand the stem cell factor to bring these two molecules together. However, during mutation in the cancer cells, they can form dimers. This is the dimers. It's no need the ligand. So this also drive the diseases to the next phase. So we know there are many natural dimeric targets in the nature. So what kind of reagents are available? So for those proteins, mostly are monomer forms on the market. Those monomer proteins, first is lost the quaternary structure conformation for the dimer proteins, and also this limited its function and also structure fidelity. There's another form of dimer proteins, that's the FC fusion dimer. This one also has its limitation because the FC is a large sequence. There has about 250 amino acids on one chain. If the two chains together will be about 50 kDs, 50 kD. This very bulk tail can potentially impact the target sequence, and especially for the smaller proteins. And also the FC has glycosylation. Plus the large molecule and the glycosylation can potentially cause masking of the critical epitopes on the target. Also, FC, we know FC can bind to FC receptors. That can potentially cause unnecessary FC related activation and also assay noises. To solve this problem, what we did at Conigen, we designed a novel dimeric motifs. This motif is a very compact size, very small, less than 50 amino acid, and also FC-free, glycan-free. Basically, you will not covering any of the epitopes. So we have demonstrated that this dimeric protein platform can produce native structure fidelity and also enhance the functional activity. So here, I will use 3 targets as examples to elaborate the dimeric protein design, immunogenicity, functional activity, and also application for in vitro assays. The first target here, I'm using the interferon alpha receptor 1 and 2 as an example. So this receptor can bind to the type 1 interferon to trigger the downstream signaling and cascade all the functions. So to design the heterodimer protein to mimic the natural conformation on the left side to show both the receptor 1 and the receptor 2, we link them together to form this dimer. So after this design, we also to see whether this design can have any potential potential impact to the structure. We modeled the structure and superimposed with the known crystal structure. The result demonstrates that the heterodimer protein can superimpose with the experimental crystal structure very well, have the high structure fidelity. So for proteins, the other thing is the quality and the bioactivity and also antigenicity. So after we produce the protein and also to analyze the protein and reducing and non-reducing condition by SDS page, you can see very high quality of dimeric proteins produced. So for the protein antigenicity, there is one important thing is bound to the specific antibodies. On the red panel, you can see the dimeric protein bound to specific antibodies very well and very highly potent. The most important, interesting thing is the middle panel here. So the orange curve is the heterodimer protein, including both receptor 1 and receptor 2. The blue and the black curves are the monomeric proteins. What you can see here is the dimeric interferon alpha receptor binding to the type 1 interferon about 50 to 100-fold increase the binding affinity. So this means this antibody, this heterodimer protein is highly bioactive. So the next one to see the dimer protein, whether this is immunogenic, to test the immunogenicity. We use the dimer protein with adjuvant to immunize mice, generate a very high titers of antibody in mice. Then furthermore, we use the hybridoma technology, isolated monoclonal antibodies. This group of antibodies, you can have different specificities. The A panel is the specificity only bound to the heterodimer protein. The B panel antibodies bound to the receptor 1. The C panel antibody bound to receptor 2. The most important information here is the antibodies only bound to the heterodimer. That means this antibody is bound to potentially the interface between receptor 1 and receptor 2. That's a quaternary structure. And also, if you see the antibody diversity, they are very diverse. So to showing that the dimeric proteins we produce is very immunogenic. The next target used here is the CD4 molecule. CD4 is the core receptor on the T cells, and also is a receptor for IL-16 and also primary receptor for HIV infection. So this CD4 was originally discovered in the early 1980s. In the later 1980s, already have publications demonstrate that CD4 can exist as monomer or dimers on the cell surface. Different cells can have different presence of the dimer or monomer. So based on the many studies for immunology studies that indicate that the dimer, CD4 dimer, is very important for the functional activity for the T cell activation. So we know CD4 is the primary receptor for HIV infection, for HIV entry. So the very recent studies to study whether one CD4 molecule or two CD4 molecule binding to the envelope protein, how that contribute to the infection. The interesting thing is that in the middle panel, if the HIV envelope protein bound to one CD4 molecule will not be sufficient to open up the envelope to bind to co-receptor or cause the fusion and viral entry and infection. So when the envelope protein bound to CD4 molecules have sufficient opening of the envelope protein for the next stage conformation change and bound to co-receptor and the viral entry. So that means this is CD4 dimer is very important for the HIV infection. So we generate CD4 dimer, the cis dimer protein. And then first thing we evaluate whether the CD4 protein can bind to HIV envelope proteins. It's very surprising to see the CD4 dimer protein bound to GP120 from different subclasses have average or on average tenfold increase compared to the monomers. The orange line is the dimer protein, the black line is the monoprotein. This is bound not only to one envelope, it's the envelope proteins from multiple subtypes. The same result was we also tested the bindings using label-free assays. This is an important application to use the dimeric protein to put on the chips to test the binding of the envelope proteins. The similar result as the ELISA results. The dimer has enhanced the binding to the envelope proteins compared to the monomer. So the binding is good bioactivity, and also more importantly to see what's the functional activity of the CD4 dimer. We collaborate with a professor at Boston University to test the neutralization against HIV-1 tier 2 viruses. For who work on the HIV knows the tier 2 virus are the very difficult viruses to be neutralized. So the CD4 dimer compare with a known CD4 molecule, CD4IG. This CD4IG has been used in the field for many decades. So when doing the neutralization against the tier 2 virus, all the viruses can be neutralized by the CD4 dimer protein, but only about 50% of viruses can be neutralized by the CD4IG. If you see the example panels, the orange curve versus black curve, also the CD4 dimer can have more potent neutralizing activities compared to CD4IG, about over tenfold increase. So this indicates that the dimeric protein not only increase the binding, but also increase the functional activities. So we also use the CD4 dimer protein to detect the CD4 specific antibodies. In this study, we collaborate with a professor in medical school or University of South Carolina. She has a cohort of HIV infected patients. This is called long-term non-progressive. Means this patient does not have a viral detected. However, this patient also have a low CD4 count. In general, if no viral detected, the CD4 cells can bounce back to close to normal level. However, in this cohort of patients, the low CD4 count. Her hypothesis is that they are autoimmune antibodies against the CD4 to suppress the CD4 cells. So when they use other forms of CD4 molecules to detect the patient's sera, they could not detect the bindings to those proteins. So then use our Conigen CD4 dimer protein and identified at least 2 patients has a positive binding to the CD4 proteins. This finding is very important to show the dimer protein can potentially increase the antibody binding. Another set of data I'm not showing here is showing the CD4 dimer protein can bind to the therapeutic antibody, calciumab, much better than the monomer protein. So this group of researchers, they will use the CD4 dimer protein to sort out B cells from the patient PBMCs to generate antibodies to do more research. The third molecule is the CD19. The CD19 is a B cell co-receptor. It has different functions to activate the B cells, including TCR dependent and the TCR independent activation functions. For the TCR independent functions, those are 2 CD4 molecules with other receptor-related molecules, for example, CD80, to activate the B cells. And also for lymphoma and the leukemia cells, the CD4 molecule intended to have a clustering. Due to clustering, there's a phase of dimerization. So therefore, we developed a panel of CD19 dimer proteins. This CD4 dimer proteins is tested for in vitro assays. One of the example is to test the binding to the CD19 specific CAR on CAR T cells. Here is the CD19 mouse CD19 CAR detection. Use the dimer protein can very sensitively to detect the CARs on the CAR T cells. But the percentage of the CAR, you can see they can detect about 15% the transfected cells, T cells producing the CAR. The network control, is, of course, is very low background. So on the next section, I'm going to talk the multi-pass transmembrane protein targets. We display this on the nanoelectric particles of the full-length proteins. The multi-transmembrane proteins are even more challenging targets and more complex proteins. For antibody discovery, we need good antigens. However, it's very difficult to isolate the pure stable multi-transmembrane proteins. And also this protein, because the extracellular domains is very small or it's complex, the immunogenicity is low. And also the conformation status is not very well studied. We show a few examples to see the antibody binding to different multi-transmembrane proteins. One is the Claudin 4 as an example, showing that the antibody Fabs only bound to one of the extracellular loops. Another example is the apelin receptor is a GPCR. The antibody is a nanobody, single-chain antibody. Those antibody can interact with multiple extracellular loops. And also the third example is a GPCR, GLP-1 antibody, also a GPCR target. The antibody Fabs can bind to the N-terminals and the multiple loops. So this also increases the complexity to display or produce antigens for antibody discovery. So Conigen has developed a technology to express the multi-transmembrane domain proteins very like a particle. So this one with higher target intensity and also better viral particle release to increase the productivity. So this is all produced using mammalian cell expression. The multi-transmembrane proteins are embedded in the natural lipid bilayer. So very similar to the proteins on the cell surface. So here we use example is CXCR4. CXCR4 is a GPCR chemical receptor. There are two ligands. One is a chemical ligand, CXCR12, and also it's also the core receptor for HIV infection. So after we produce this CXCR4 full-length VLP, and first tested the binding to the specific antibodies, very potent bond to the antibodies. The EC50 is in the single-digit nanogram per ml, very potent. And more importantly, to showing that these GPCRs on the cell surface can bind to its ligands. So HIV, GP120 protein, and its chemical ligand, CXCR12. The result that showing that indicate the CXCR4 on the cell surface mimic the natural conformation, can bind to their ligands and also their potent bound to antibodies. These are bioactive. The other example is the CXCR5. CXCR5 is also a GPCR chemical receptor. The CXCR5 viral particle, we detected the antibody binding against the specific antibody very potent. Also, the binding to the antibody is in the single-digit nanogram range, and also have a potent binding to its ligand, CXCL13. The further demonstrate that the Conigen's multi-transmembrane domain protein VLP platform can generate viral particles with target that are bioactive, mimic the natural conformation. So this gives you a summary of the toolboxes generated in Conigen. That's the protein toolboxes for antibody discovery, though including the soluble dimer proteins and the full-length multi-transmembrane proteins viral particles. So if you look at antibody discovery stages from the target validation, antigen design, we all can contribute to this to design the desirable antibody geneticity to have good antibody, good antigen epitope presentation. The second box showing the immunogenicity. The goal is to generate immunogens with a strong immunogenicity to induce desired antibody responses and high-quality antibody responses. The third bottom, the third box showing the antigens can be used for many different kinds of in vitro assays, including antibody screening, selection, validation. So those showing this antigens can present the correct antibody for antibody discovery. The next section, I'm going to talk about the novel fit for target rational design for immunogens and also for immunization strategies. So that will give, based on each target, we'll design different forms of antigens. The goal is to generate antibody responses in the immunized host and break immune tolerance, generate high quality antibody responses. As I mentioned, the antigens for therapeutics, majority of endogenous antigens. These are low immunogenic. So how to generate antibodies, we need to first break immune tolerance and also generate high quality B cells antibodies. So this one, as I mentioned, we will do very extensive antigen, the protein sequence analysis to generate the good, suitable immunogens. And also, based on the target need, we can design multiple forms of antigens, including potential loops or conformational epitopes. And then select the immunization design to fit for the target formulation and also immune focus to generate antibody responses. So here we only show the examples of GPCR targets. We have developed antibodies against a broad range of GPCR targets. Here are a few examples here. So the antibodies not only bound to the protein, but more importantly, can bound to the target on the cell surface by flow cytometry. So these targets you can see covered many of the class A molecules, class B molecules targets, and also class C and F targets. Here, I'll just show you a few examples of the GPCR specific antibody responses in mice against different variety of GPCRs, class A, B, and C and F. If you look at the histogram, the GPCR specific antibodies in the mouse serum very potent to bound these targets. This is a very good sign for next step to develop monoclonal antibodies. So this is the slide I showed earlier to show their broad range of GPCR targets. We have general antibodies that can bind to the cell surface. So then we use the monoclonal antibody technology, hybridoma technology, to generate antibodies, monoclonal antibodies from the immunized mice. So these are a few examples of the monoclonal antibody we generate against the class A, B, and F targets. So these targets are important therapeutic targets, DRD2, PAR1, GLP1R, GIPR, and FASO10. So in conclude, this one, the Conigen's proprietary immunization platforms are very efficient to generate antibodies in mice against those very challenging targets. In summary, the take-home message is the first is the conformational proteins are very important, can be very useful for antibodies discovery success. And we have two different forms of proteins, the soluble dimeric proteins, and also very easy to use as an immunogen for immunization, and also as antigen for all kinds of in vitro assays, and also to fish out antibodies specific B cells. Similarly to the multi-transmembrane protein on the viral particle, those are bioactive and mimic natural conformation in the natural lipid bilayer. The third platform is the precision antibody platform. We have successfully generated antibodies against the difficult targets to break immune tolerance and also generate monoclonal antibodies. So Conigen is not a therapeutic company. We can have multiple mechanisms to support the antibody discovery. We all have the catalog of products ready to the community. And also we can have customer projects to work with you to develop conformational proteins or immunization or antibody discovery against a specific target. The collaboration and partnership, we also can have different mechanisms to work with academia, biotech, and pharma. So it's very important to have a great team, very intelligent, skillful scientists, product management, production management at the Conigen. So they all contribute to the success of the Conigen's platform building and the product production and also antibody generation. I also thank the collaborators from Boston University and Medical School of South Carolina and Gates Bell. The other very important thing is the Conigen is one of the companies founded by the Conifer Life Science Group, second them all. Thank you for your attention. Thank you for your participation of this webinar. If you have any questions, we will try to answer. If we cannot answer here, please feel free to email me and we can answer later. Thank you.